Hi students, today's topic is very important topic from carbohydrate metabolism, that is gluconeogenesis. This is the only question where you won't get any clinical scenario or clinical case history like that. Direct essay question, gluconeogenesis. The name itself, the definition for gluconeogenesis is gluconeogenesis, that is production of new glucose, right? From the non-carbohydrate substances. If the glucose is available means there is no place for gluconeogenesis. If there is no glucose means our body will try to synthesize glucose from available non-carbohydrate substances. This is very important, right? So definition is synthesis of glucose from non-carbohydrate substances. Second one is, as usual, what is the site and location? Cytosol and mitochondria. In both compartments, the gluconeogenesis will happen. Location is usually two organs, that is liver and kidney. Sometimes some amount of gluconeogenesis takes place in intestine also. What are all the significances? We know the first reaction gives energy is glycolysis. Second is glycogenolysis, both from liver. Third one is gluconeogenesis. Actually, without glucose, this one gluconeogenesis will coming for the rescue of blood glucose. Second one is up to 12 hours or 18 hours glycogenolysis will take place. After that, gluconeogenesis will come for the supply of glucose. Right. What is the significance of this pathway? Number one, 24 hours glucose is to be supplied for our brain. That needs 24 hours glucose. If there is any gap for the glucose supplied to the brain, this gluconeogenesis will take over the steps and continuous supply of glucose to the brain. Second one is, this gluconeogenesis only maintain the blood glucose regulation. So 2 points for the significance. Okay. Next, we are telling in the definition non-carbohydrate substances. What are all they? Propionyl-CoA, alanine, that is amino acid alanine. Third is lactate. Fourth one is glycerol. In another video, I will explain all the four substrates, how finally they will give glucose and maintains the blood glucose level. Okay, but we should know about the four substrates name. Propionyl-CoA, that is short chain fatty acid. Lactate, that is end product of anaerobic glycolysis. Alanine, that is glucogenic amino acid, and the last one is glycerol, which is derived from fatty acid, that is triacyl glycerol. Okay. Yes, the name of the substrates are four, that is propionyl-CoA, glycerol, lactate, and amino acid, that is glucogenic amino acids, alanine, and also glutamine. Alanine in liver, glutamine in kidney. So now we are going to discuss about how these four substrates will give glucose for the fasting state or in the starvation state to maintain the blood glucose, right? The first one is glycerol. Actually, it is a waste product from lipid metabolism. After lipolysis only you will get glycerol. After hydrolysis of TAG, that is triacyl glycerol, after hydrolysis only will release glycerol, right? This glycerol is converted into glycerol 3-phosphate with the help of glycerol kinase. This is a very important MCQ point, because glycerol kinase is not present in adipose tissue. This is present in liver and kidney only. This glycerol 3-phosphate again converted into dihydroxyacetone phosphate. We already discussed in the metabolism glycolysis. Usually glycerol 3-phosphate is converted into dihydroxyacetone phosphate, which enters into the pathway of gluconeogenesis and then produces glucose finally. So this is how the glycerol is synthesizing glucose in gluconeogenesis. The two organs are liver and kidney only, but in adipose tissue there is no glycerol kinase, so no place for gluconeogenesis. Second one is lactate. You all know the end product of glycolysis is pyruvate in aerobic conditions. In anaerobic only the lactate is produced. This lactate is actually in the situation of strenuous exercise or vigorous exercise in the muscle only, they will go for anaerobic glycolysis and produces lactate, right? In muscle, this glucose produces pyruvate. Pyruvate is converted into lactate with the help of lactate dehydrogenase enzyme, right? This lactate enters into the liver through the bloodstream, that is in between blood. Through the bloodstream, they enter into the liver. In the liver, the lactate is converted into pyruvate. After that, this pyruvate only enters into gluconeogenesis, finally produces glucose. You all know that TCA cycle, final end product is oxaloacetate. That oxaloacetate is synthesized from pyruvate. This oxaloacetate is present in mitochondria, which is shifted to the cytosol and produces phosphoenol pyruvate. These are all the glycolysis steps. But you should know the lactate is converted into pyruvate. This pyruvate again enters into gluconeogenesis pathway, produces finally glucose. This glucose is again transferred to muscle for their energy. This is the Cori cycle, that is C-O-R-I, Cori. It is nothing but transfer of lactate from muscle to liver, known as Cori cycle. Okay, how the pyruvate is converted into lactate and finally how the lactate is converted into pyruvate, finally produces glucose is the concept of Cori cycle. Third one is alanine. There are so many glucogenic amino acids are there. Usually alanine will come for the gluconeogenic pathway in the fasting or starvation situations. When you will get alanine? Breakdown of protein and dietary protein. This alanine synthesized in muscle. The reaction name is transamination. See, in the muscle, glucose converted into pyruvate. Pyruvate is again converted into alanine. How? Alpha amino acid going for transamination reaction finally produces alpha keto acid and alanine. This is one transamination reaction occurs in muscle that produces alanine. This alanine enters into the liver. Why? Again they will go for transamination reaction, that is alpha keto acid combined with alanine produces pyruvate and glutamate. This pyruvate again enters into the gluconeogenesis pathway, finally produces glucose. So our concept is, with the available non-carbohydrate substances, try to bring out in the gluconeogenesis pathway, finally produces glucose. Okay. So in the liver, this glucogenic amino acid alanine enters into the transamination reaction, converted into glutamate and pyruvate. Pyruvate enters into gluconeogenesis pathway and finally produces glucose. This is the third non-carbohydrate substrate. The last one is propionyl-CoA. This is best example for odd chain fatty acid. Propionyl-CoA is, after oxidation of fatty acids, you will get propionyl-CoA. This propionyl-CoA converted into D-methyl-malonyl-CoA and L-methyl-malonyl-CoA. This reaction is very, very important because the enzyme name is also important, the coenzyme name is also important. So we need three molecules, one molecule of CO2, one molecule of water, one molecule of ATP for the conversion of propionyl-CoA to L-methyl-malonyl-CoA. The enzyme name is propionyl-CoA carboxylase biotin, that is carboxylation reaction vitamin. Biotin is the coenzyme for this reaction. This methyl-malonyl-CoA finally converted into succinyl-CoA with the help of methyl-malonyl isomerase. This is the enzyme name. For this isomerase, we require vitamin B12, right? Then only the reaction is converted and produces succinyl-CoA. You all know about the fate of succinyl-CoA. Enters into TCA cycle, and this TCA cycle, that is oxaloacetate, up to oxaloacetate the TCA cycle will occur. Then this oxaloacetate will be converted into phosphoenol pyruvate and finally produces some amount of glucose through gluconeogenesis. So this is how propionyl-CoA enters into TCA cycle and finally produces gluconeogenic pathway glucose. Okay, so these are all the four substrates for, that is non-carbohydrate substrates for gluconeogenesis. Yes. Now the next step is for this gluconeogenesis pathway. You have to draw the pathway very carefully because this gluconeogenesis takes part in both compartments, cytosol and mitochondria. So what is the first product, initial product, how it enters into the, where it is synthesized like cytosol, how it enters into mitochondria, again how it comes for the cytosol and proceed the synthetic pathway. Like that you have to draw the pathway very carefully. I will explain in the next part. So in this pathway you have to mention mitochondrial reactions and cytosol reactions. And for this pathway, before that, to draw the pathway, you should know about the malate aspartate shuttle. With this help of this shuttle only, the substrates are transferred from cytosol to mitochondria or mitochondria to cytosol. That is malate aspartate shuttle. Okay. Next one is how it is regulated. As usual, we are having the same 3 points. Number one, substrate availability. If the glucose is available means they will go for glycolysis. If the glucose is not there, not available means the reaction name is gluconeogenesis. Second one is we have 4 pathway enzymes are there. One is pyruvate carboxylase. Second one is phosphoenol pyruvate carboxykinase. Third one is fructose 1,6 bisphosphatase. The last one is glucose 6 phosphatase. Right? You have to mention in the pathway very carefully. Next one is we have 2 hormones, that is hypoglycemic and hyperglycemic. You know the only hypoglycemic hormone is the insulin. The other hyperglycemic group comes glucagon, glucocorticosteroids like that. So this insulin decreases or inhibits the gluconeogenesis, whereas glucagon and glucocorticoids stimulates the gluconeogenesis depending upon the supply of glucose. It's now coming to the energetics. In metabolism biochemistry, energetics means either you have to give some ATPs to synthesize the products, that is utilization of ATPs. Another one is from that pathway, specific pathway, we are getting some ATPs, that is production of ATPs, right? These 2 points are coming under energetics. It's a common topic. In this gluconeogenesis, energetics means utilization only. That is, we are giving 6 molecules of ATP for the production of 1 molecule of glucose, right? That is why we have to give 6 ATPs for the production of gluconeogenesis 1 molecule. Whereas from the glucose by anaerobic glycolysis, that is lactate end product, this pathway we are getting, that is they are synthesizing 2 molecules of ATPs. These 2 points are very, very important MCQs. In glycolysis, anaerobic glycolysis, ATP production is 2. Whereas in gluconeogenesis, you have to give 6 ATPs for the synthesis of 1 molecule of glucose. Okay. And the final topic is subtitle is disorders. Is there any disorders involved in gluconeogenesis? Yes, we have disorders. That is lactic acidosis, that too high anion gap metabolic acidosis. Second one is ethanol poisoning. Third is halothane, that is anesthetic drug. Both they have inhibited the gluconeogenesis, and last one is pyruvate carboxylase deficiency. These 4 are the very, very important MCQ points. Number 1, very rarely they are asking any short notes regarding disorders of gluconeogenesis, right? So in this gluconeogenesis, you just see 1, 2, 3, 4, 5, 6, 7 short notes are there. That is why gluconeogenesis is very, very, very important essay question from carbohydrate metabolism, right? Yes. Now we are going to draw the pathway. Very important step for gluconeogenesis. How to start, where to start, how it finish, where is the final end product, where is the initial product. Very important points. Labeling. For gluconeogenesis pathway, first you have to start with pyruvate. Right? We know the pyruvate is synthesized from cytosol in glycolysis. Right? Cytosol in glycolysis, that is before the mitochondria, from phosphoenol pyruvate. Here the enzyme is pyruvate kinase, right? This is from glycolysis. Now the point is, from the cytosol, the pyruvate is transported into the mitochondria. You got it? Now the pyruvate is available in mitochondria for the further reactions. Okay. Usually what happens in glycolysis, the glucose is available very much. Pyruvate is automatically converted into acetyl-CoA with the help of pyruvate dehydrogenase enzymes. Here what happens is there is no glucose. So this pyruvate, that is available glucose, produces pyruvate, which will enters into the TCA cycle, then produce some products, substrates, that will again go to the cytosol, finally produces glucose. This is the concept of diagram. So this cytosolic pyruvate enters into mitochondria, number one. Number two, this mitochondrial pyruvate is converted into oxaloacetate because there is no glucose supply. This oxaloacetate is converted into malate. These two are the products of TCA cycle. This malate is transported to the cytosol through malate aspartate shuttle. That is why for gluconeogenesis, don't forget to mention or don't forget to draw about the malate aspartate shuttle path. Right? So through this malate aspartate shuttle, the malate is transported from mitochondria to cytosol. That is mitochondrial membrane, out of the mitochondria, cytosol. This malate is again converted into oxaloacetate. This oxaloacetate is converted into phosphoenol pyruvate with the help of energy molecule GTP. Very important point. For the conversion of phosphoenol pyruvate from oxaloacetate, we require one energy molecule GT. Right? This is the first part of your diagram of gluconeogenesis. Usually what happens? Oxaloacetate produces citrate, citrate produces alpha-ketoglutarate, then succinyl-CoA, fumarate, malate, like that. Why I am drawing this TCA cycle in this area is, we already know the substrate, that is glucogenic amino acids, enters into TCA cycle through alpha-ketoglutarate. Propionyl-CoA converted into succinyl-CoA enters into TCA cycle. Glucogenic amino acids again enters into TCA cycle through fumarate. So these are all the compounds which receives the non-carbohydrate substrates for the production of glucose. Okay, so this is the first step. Actually, lactate is another substrate which will be converted into pyruvate, again enters into gluconeogenic pathway, finally produces glucose. That is why lactate is here. Glucogenic amino acids are there. Propionyl-CoA is there. Next, how to extend the diagram or how to complete the diagram. So your next available, that is resting product, is phosphoenol pyruvate with the help of GTP. This phosphoenol pyruvate automatically produces glucose by the reversal steps of glycolysis. Okay, so don't worry about this. 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bisphosphoglycerate, and glyceraldehyde 3-phosphate. These are all the routine steps in glycolysis. Here, one molecule of GTP is required for phosphoenol pyruvate. Another molecule of ATP is required for this pyruvate carboxylase. One more ATP is required for the conversion of 3-phosphoglycerate to 1,3-bisphosphoglycerate. Then the product is glyceraldehyde 3-phosphate. You know how it will reversibly changed or irreversibly changed. It is always reversible reaction converted with the dihydroxyacetone phosphate. Here the last non-carbohydrate substrate glycerol enters into gluconeogenic pathway through dihydroxyacetone phosphate. Again the reaction continues, fructose 1,6 bisphosphate, then fructose 6-phosphate. Here the enzyme is fructose 1,6 bisphosphatase. Then fructose 6-phosphate again converted into glucose 6-phosphate. Finally they will produce glucose with the help of glucose 6-phosphatase. This is how you have to draw the diagram. Don't start from the glucose molecule. Take one full page. Middle of the page you just mark the mitochondria. Before mitochondria cytosolic pyruvate, after mitochondria mitochondrial pyruvate. Then this mitochondrial pyruvate how converted into oxaloacetate, malate, and finally produces cytosolic phosphoenol pyruvate. And second point is how the oxaloacetate continues the TCA cycle to receive the glucogenic amino acids and propionyl-CoA R chain fatty acids like that. Then this phosphoenol pyruvate continues the reversal of glycolysis reactions with one or two exceptions. This is exception, this is exception. Finally, one molecule of glucose is produced by the whole synthetic gluconeogenic pathway in the two compartments. First start with cytosol and ends with continuous mitochondria, finally ends with cytosol. Okay.